Hello, everyone. Welcome to today's ASBMB Breakthrough webinar. We have a really nice talk for you today. My name is Isabel Casas. I'm the Director of Publications for ASBMB Journals. And today's webinar is brought to you by the JBC, the Journal of Biological Chemistry. A few reminders before we dive in. You will have questions at the end. Please go ahead and submit them in that Q&A box at the bottom of your screen. This webinar is being recorded and is going to be available on our ASBMB website. Without further ado, it's my pleasure to introduce today's speaker, Alex Toker. He's a professor of pathology at Harvard Medical School and the JBC editor-in-chief. So please join me in welcoming Alex. Alex, the floor is all yours.
Thank you, Isabel. Thank you all for joining. It's a pleasure to be here with you today to give this ASBMB Breakthrough webinar on the discovery, regulation, and function of the PI3 kinase and AKT pathway in cancer, a topic of research that has occupied my interests in my laboratory research for the better part of a quarter century. Happy holidays to you all. This is my disclosure slide. The most relevant and important thing is that I am the editor-in-chief of the Journal of Biological Chemistry, and we do have collaborations with a number of companies who support our research and some of their products I will be discussing today. I will divide the webinar in two major areas. One is an introduction to PI3 kinase, AKT, mTOR, and PTEN signaling, and the second part focusing on some of our recent advances in identifying targetable vulnerabilities for therapeutic benefit in patients that have hyperactivation of this pathway in the context of cancer. So just some basics to bring everybody onto the same page. PI3 kinase AKT, as illustrated in the signaling diagram, is a classical lipid-derived signaling pathway that responds to extracellular cues in the form of growth factors or hormones that converge on the activation of receptor tyrosine kinases or G-protein coupled receptors that through a complex mechanisms and signal relay enzymes converge on the activation of a family of lipid kinases, collectively known as the phosphoinositide 3 kinases. The one that I'm going to be talking about today is phosphoinositide 3 kinase that interconverts a minor membrane phospholipid, PI3K, into a product, phosphatidylinositol 3,4,5 trisphosphate, or PIP3 for short. This is a true second messenger. It fulfills all of the requirements of bona fide second messengers. It is nominally absent in unstimulated cells. It rapidly accumulates at sites of synthesis. And it is equally rapidly degraded by active mechanisms. So if PI3 kinase carries out the forward reaction, the lipid phosphatase PTEN interconverts it back into its substrate. Once PIP3 accumulates at the plasma membrane, it engages downstream effectors. And the subject of today's talk is going to be on the serine-screening protein kinase, AKT, which is a downstream effector of PI3 kinase in all cells and tissues. This slide also depicts the fact that although most of the activity occurs at the plasma membrane, there is a pool of PI3 kinase, its lipid products, and downstream effectors found at endomembranes. Once activated, AKT transduces the signal by phosphorylating a number of downstream substrates shown here. There are currently about 200 or so substrates that have been identified as AKT substrates that are phosphorylated in a given cell or tissue. And as we argued in this review that Brendan Manning and I wrote in 2017, there are three multifunctional, evolutionarily conserved nodes: the 4-kidboxo family of transcription factors, the glycogen synthase kinase 3 enzyme, and the tuberous sclerosis protein of the TSC complex. Phosphorylation of these proteins by AKT serves to inactivate their localization or function, and thereby allowing them to transduce a signal to phenotypes that include cellular survival, proliferation, metabolism, growth, and other cellular responses, all of which are corrupted in pathophysiology such as cancer. There are, of course, many, many more substrates beyond FOXO, GSK3, and TSC2, and some of them are depicted down here. These serve to transduce signal relay to these various phenotypes. I do want to spend a minute highlighting the notion that there is a lot more to PI3 kinase signaling than just AKT. We just understand it very poorly. There are related kinases, such as the serum and glucocorticoid kinases, depicted here, that also sense the PI3 kinase and PIP3 signal and transduce the signal to overlapping cellular responses to those mediated by AKT. There are tyrosine kinases, such as BTK, Bruton tyrosine kinase, which also has lipid binding domains, such as the pH domain, that engage PIP3 at the plasma membrane and transduce the signal through a complex series of reactions to cellular responses, such as adhesion, migration, and cellular proliferation. And finally, there's a whole family of PIP3 sensitive guanine nucleotide exchange factors for small GDPases, such as RORAC and CDC42. These also sense the PIP3 at the plasma membrane, and these transduce signals primarily associated with remodeling of the actin cytoskeleton, leading to changes in cell migration, invasion, as well as metabolism. We understand the functional regulation and role of these non-AKT effectors a lot less than we understand the mechanisms that account for PI3 kinase AKT signaling, both in terms of cellular physiology as well as pathophysiology. One of the reasons, or primary reason, why this pathway has gained so much attention in the oncology world is because this is, in aggregate, the most frequently mutated pathway in all cancers, in all solid and hematological malignancies. Genes whose protein products participate in signal relay through PI3 kinase, in aggregate, are the most frequently altered genes in human cancers. If you query TCGA, ask questions of the mutational status of PI3 kinase pathway genes at the level of somatic mutation, gene fusion events, genetic amplification and copy number gains, as well as deep deletions in tumor suppressors, upwards to 70 to 80 and sometimes even 90% of patients exhibit one or more, typically, genetic alterations in the PI3 kinase pathway genetic makeup. Just to focus on this a little bit more, this is primarily due to the fact that PI3 kinase itself, the PI3 kinase alpha subunit encoded by the PIK3CA gene, is an oncogene, as first identified by Bert Vogelstein and Victor Valculescu at Hopkins, who identified frequent hotspot mutations in the PIK3CA gene in human cancers. As originally recognized by John Carpton and Jim Thomas at Eli Lilly, there is a somatic activating mutation in the AKT1 gene that renders it oncogenic, weakly oncogenic, but oncogenic nonetheless. And then finally, in the late 1990s, two groups independently identified PTEN. This is the enzyme that interconverts PIP3 back to its substrates and therefore puts the brakes on PI3 kinase and PIP3 signaling. Therefore, loss of this gene would promote constitutive activation of the pathway. It was Ramon Parsons at the time at Columbia who, through positional cloning, identified a region on chromosome 10 in glioblastoma patients that was subject to frequent loss of heterozygosity. And it bore the gene, the open reading frame, bore similarity to protein tyrosine phosphatases. And subsequently found that it was frequently lost by LOH in brain, breast, prostate, and other carcinomas. At virtually the same time, Jack Dixon's lab at the University of Michigan was doing protein purification of tyrosine phosphatase activities and cellular extracts and found a novel activity that, instead of dephosphorylating tyrosine residues, actually dephosphorylates PIP3, interconverting it back into PI45P2. And it turned out that this protein lipid phosphatase activity is PTEN, a JBC rapid communication published by Jack in 1998. Suffice it to say that PIK3CA somatic mutations are number 2, second only to KRAS, in terms of frequency of genetic alterations across all human cancer lineages. PTEN is the most frequently inactivated tumor suppressor across all cancer lineages, second only to P53. It's also worth noting that there are 3 AKT isoforms in mammals and humans, which reside on different chromosomes. They have remarkable similarity or identity in the 3 major regulatory domains, the pH domain, the catalytic domain, and the hydrophobic tail. And although originally thought to have redundant functions, at first really educated to us by Maury Birnbaum, who made the first AKT1, AKT2, and AKT3 whole animal knockout mice, they actually have non-redundant and specific functions, both in normal cellular physiology as well as in disease. And our lab, as well as Jabrugi's labs and many other labs, subsequently went on to find that AKT1 and AKT2 in particular have non-overlapping distinct functions in the context of cancer etiology, progression, and metastatic dissemination. It was Luke Anthony's group back in the 1980s, along with two other labs, who independently discovered a PI3 kinase activity in cellular extracts, as first published by Lou in this 1985 Nature paper. But especially for the trainees on the call, bear in mind that it took 40 or so years of discovery science, research, drug development, preclinical models, and clinical trials for the approval of the very first PI3 kinase alpha-specific inhibitor, alpelisib, developed by Novartis, to be approved by regulatory agencies such as the FDA. This approval came in 2019, where it was shown that alpelisib, this PI3 kinase alpha-specific inhibitor, in combination with endocrine therapy fulvestrant in hormone receptor-positive breast cancer patients, HER2-negative, showed a doubling of progression-free survival compared to patients treated with placebo plus fulvestrant in pathway-altered tumors. That is to say, HR-positive breast cancer patients with either PIK3CA mutation, AKT somatic mutation, or PTEN inactivation. By the same token, it took about 35 or 36 years for the first AKT inhibitor, capivasertib, an AstraZeneca competitive catalytic inhibitor, to be approved by the FDA, again in hormone receptor-positive, HER2-negative breast cancer patients with pathway alterations in combination with fulvestrant. This took 36 years since the original discovery of the AKT1 and AKT2 genes by Stephen Staub at Hopkins in 1987 in this PNAS paper. I want to show you a timeline of the development of these drugs over the years. So back when I was a postdoc, back in prehistory, we only had real tool compounds, such as these drugs known as vortmananil, Y294002. Now, although these had terrible solubility, toxicity, and off-target effects, they were instrumental for discovery science in both PI3 kinase and AKT signaling. But it wasn't really until the 2010s and the late 2010s when the first PI3 kinase specific inhibitors were developed and ultimately approved. The very first one was idelalisib, which is a PI3 kinase delta inhibitor, approved for chronic lymphocytic leukemia. And as I just mentioned, alpelisib was approved in 2019, a PI3 kinase alpha inhibitor for hormone receptor-positive breast cancer patients. Fast forward to the present day, about a year ago, the FDA approved another PI3 kinase alpha selective inhibitor, again for hormone receptor-positive, HER2-negative breast cancer patients with pathway alterations in combination not just with fulvestrant, but the CDK4/6 inhibitor palbociclib, triple combination. With AKT, it's been about 5 years behind. Back in the 1990s, we had very poor lipid mimetic inhibitors, but these were great tool compounds for discovery science. The first real proper inhibitor came along, was developed by Merck, so MK2206. But this showed no benefit and prohibited those limiting toxicities as single agent in clinical trials. It wasn't until 2023, as I just mentioned, that the AstraZeneca compound capivasertib was trialed and approved again in hormone receptor positive, pathway altered breast cancer patients. And if you fast forward, and this is what I'm going to spend the rest of the talk today about, is where are we going today and in the future? We now have the development of antibody-drug conjugates. I'm going to show you some of that. We have degraders targeting PI3 kinase. We have mutant-selective inhibitors. And the same is true for the AKT arm of the pathway. We have ADCs, PROTACs, and isoform-selective inhibitors. Why do we need better inhibitors? Why are the ones that have been approved to date, both PI3 kinase alpha and AKT inhibitors, why are they not good enough? Well, the problem is that patients only derive a small amount of benefit on these agents, which are used typically as third-line therapy and only in hormone receptor-positive breast cancer. And the reason is because these drugs suffer from dose-limiting toxicities, notably hyperglycemia, hyperinsulinemia, and rash. And many patients in the real world simply do not tolerate these drugs. Also, these drugs initiate almost immediately resistance mechanisms, bypass mechanisms that allow the tumor to regrow. None of these drugs on the AKT side of things are isoform selective. And AKT does have scaffold functions that we understand very poorly, and catalytic inhibitors will simply not inhibit these activities. So for the rest of the time, I'm going to talk about 4 major areas of research that are all ongoing in our lab and many other labs around the world. Begin with antibody-drug conjugates. The challenge here is that, as with all PI3 kinase and AKT and other small molecule inhibitors in this arena, is that they display prohibitive dose-limiting toxicities, notably hyperglycemia. These can be mitigated in the oncology clinic for some patients, but certainly not all. And so the solution is to target, because these are given systemically, okay, in the form of a pill. And so they hit all organ systems in the body. And the goal here would be to target the inhibitor directly to the tumor. And one strategy would be to use antibody-drug conjugates. As you know, these are antibody-based biologics that have a monoclonal antibody, which through a chemical linker adds a cytotoxic or small molecule inhibitor payload to this molecule that can be delivered directly to a tumor that expresses the antigen to which this antibody binds. And the most successful antibody-drug conjugates that have been used and approved in the clinic are trastuzumab-based. So trastuzumab being a HER2 monoclonal antibody that absolutely revolutionized the field of breast cancer therapy in the late 1990s, mostly through the work of Dennis Slayman and his colleagues. That really revolutionized treatment options, efficacy, and provided real benefit to HER2-positive breast cancer patients. Fast forward to the 10, 20 years from then, next generation combination and resistance strategies emerged. But what really gave it a shot in the arm is the development of trastuzumab-based antibody-drug conjugates, where cytotoxic payloads were delivered through trastuzumab. There are now 7 approved ADCs for hematological malignancies. There are 8 approved for solid tumors, and there are over 100 ADCs in clinical trials. In spite of this remarkable success, there are some limitations with current ADCs. First of all, the drug-antibody ratio, DAR, is typically only 2:8. That means that there are only 2 molecules of a cytotoxic payload per monoclonal antibody. This limits the payload that can be delivered to the tumor. And patients invariably develop resistance due to the activation of compensatory pathways, including PI3 kinase AKT. So we have entered into a collaboration with a biotech company known as Nurse Therapeutics, who have developed a rather remarkable chemical technology known as HiDAR, high drug antibody ratios, ADCs. And the name is exactly what it implies. Instead of delivering 2 to 8 molecules of the cytotoxic payload per antibody molecule, they can deposit 50 to hundreds of molecules onto the ADC. And they do so because they are able to build a polymer-based backbone that is attached to the monoclonal antibody. And not only can you deliver one cytotoxic payload, you can actually decorate this polymer backbone with multiple different cytotoxic payload. And we initiated this by adding the cytotoxic payload in the form of a dual PI3 kinase mTOR inhibitor, apatalesib, which was developed about 10 years ago. Very potent, exquisitely specific, and with terrible dose-limiting toxicities. And so this fell out of phase 1 and phase 2 clinical trials simply because of that. But now, because it was given systemically, but now because we can deliver this directly to tumor cells, we have added this onto trastuzumab, and in this tumor xenograft of a HER2-positive breast cancer xenograft, the HiDAR trastuzumab outperforms the free drug apatalesib, even at doses which have dose-limiting toxicities. But most importantly, it outperforms the standard of care in the clinic, Enhertu or trastuzumab DXD. And it does so without inducing dose-limiting toxicities in the form of elevations in blood glucose in the diabetic glucose range. So if this is pre-apatalesib, the HiDAR ADC at doses which give you efficacy does not induce any elevation in the diabetic blood glucose range. So we are very excited by this technology. And we imagine by decorating these polymer backbones with distinct and different cytotoxic payloads, as well as different monoclonal antibodies that can engage different cancer lineages, we might be able to have remarkable efficacy while sparing dose-limiting toxicities. The second challenge is that patients typically develop rapidly resistance to these inhibitors, and this would include ADCs, such as the one that I've shown you. One way to mitigate this is to not just inhibit the kinase, but to degrade the protein wholesale. And this is the basis of the PROTAC, or proteolysis targeting chimera technology. This induces proteasomal degradation of a given protein using heterobifunctional polypharmacology. So a few years ago, we collaborated with Nathaniel Gray and many other scientists in our community here at Harvard, and we generated the first and second generation AKT degraders, or PROTACs, that exquisitely potently and specifically degrade all three AKT isoforms. And you can see an example of this pharmacology here. This is our first generation AKT degrader. This is our second generation AKT degrader. In a matter of hours at low nanomolar concentrations, you can degrade AKT within a few hours with concomitant inhibition of downstream signaling as measured by phosphorylation, inhibition of phosphorylation of this AKT substrate. And these are remarkably potent, and this is a complex slide, it's a proliferation assay, but all you really need to know is that this black and orange line here are our first and second generation degraders that outperform all of the currently available catalytic and allosteric AKT inhibitors that are in the clinic. Simply saying that degrading AKT is more potent and more effective than simply inhibiting its catalytic activity. And in current studies in the lab, we are trying to understand the mechanistic basis for why degrading AKT is more potent than in simply inhibiting its catalytic activity. And these are some of the degraders that we developed. We have a collaboration now with Alessia Trulli in Dundee, and this is another completely different scaffold that was developed by Ramon Parsons and Jian Jin at Mount Sinai. Where there is also a tremendous amount of excitement is in selective PI3 kinase and AKT inhibitors, mutant and allele-specific inhibitors. All of the drugs that I've shown you so far that are in the clinic inhibit both the wild type and the mutant alleles in tumors. And this is largely the reason why you have dose-limiting toxicities, because when administered systemically, these drugs inhibit PI3 kinase and AKT in all organ systems in the body, including the liver. And that's why you get DLTs. But if we could inhibit only the mutant allele or the mutant protein, we might be able to mitigate these dose-limiting toxicities. So at least 3 companies have now developed mutant-selective PI3 kinase alpha inhibitors that inhibit only the mutant oncogene in the tumor. These have been developed by Relay, Scorpion Therapeutics, and Loxo, now acquired by Lilly. And you can see in this simple experiment that this mutant-selective inhibitor outperforms and moves and shifts the curve to the left instead of expressing the mutant allele of PIK3CA while sparing cells that express only the wild-type allele. And to fast forward to the present day, there are now efforts by 4 companies, 3 of which are depicted here, and we have a collaboration ongoing with Atavistic, who have developed mutant-selective AKT inhibitors, as well as isoform, AKT1 isoform selective inhibitors that spare AKT2 and AKT3. So while none of this has actually been published, although the PI3 kinase alpha and mutant-selective inhibitors have been published, watch out for the development of these drugs as they enter the clinic and clinical trials in the next few years. And I would be remiss to not mention the Ras PI3 kinase alpha breakers, which I'm very excited about as well. It's many years ago that it was Julian Downward's lab that first identified that in a certain number of solid tumors, in particular lung cancer, the interaction of PI3 kinase alpha and KRAS is required for tumor maintenance. So there's a direct interaction between RAS and PI3 kinase alpha that is required for tumor etiology. And if you could disrupt this interaction with a small molecule, you might be able to mitigate a tumor progression. And so BridgeBio, that grew out of some of the work that followed this original work from Julian, actually developed just such a compound. This is what's known as a RAS PI3 kinase breaker, developed by BridgeBio that came out of the RAS initiative at the NCI. As single agent in a dose-dependent manner, this RAS PI3 kinase breaker not only shrinks, but actually induces tumor regression at doses that are completely tolerated and show no overt dose-limiting toxicities. There's a lot of excitement about this and other RAS PI3 kinase breakers that have now entered the clinic. The first patient of this particular PI3 kinase alpha RAS breaker was dosed about a year ago. I want to spend the rest of the next 10 minutes talking about how do we use discovery science to identify druggable, targetable vulnerabilities in the context of the drugs that I've just presented to you. Because I would argue that none of these drugs are going to work as single agent. Combinations, if we've learned anything from the last 20 years from the oncology clinic, combination strategies are the ones that are going to give the most durable therapeutic responses. And I will remind you that so far, in both PI3 kinase and AKT space, the drugs that have been developed, which are exquisitely potent and exquisitely specific, these three have only been approved in hormone receptor positive, HER2 negative breast cancer. The other breast cancer lineages have not shown efficacy, and no other solid tumor lineages have shown efficacy as well, either as single agent therapy or the few trials that have been done for combination therapy. And the poster child for this is triple negative breast cancer. TNBC has as high a frequency of PI3 kinase pathway alterations as estrogen receptor positive. And indeed, phase 3 trials with the AKT inhibitors were done. The Capitello 290 trial, the Opportunity trials in TNBC, all of these failed in combination with standard of care cytotoxic chemotherapy. They either did not meet the dual primary endpoints or failed to improve progression-free survival. Yet these same drugs not only showed benefit in hormone receptor positive breast cancer patients, they are now approved and given to patients in the clinic every day. So why is this? It's hard to know whether it's clinical trial design or something about the genetic makeup of other cancer lineages. But one way to approach this is to do discovery science. And the way we and many others have been doing this is to do synthetic lethal screens. This takes advantage of the concept that was really championed by the development of PARP inhibitors, where a combination therapy gives you synthetic lethality in the laboratory when if you inhibit one arm of a pathway, you get a cytostatic response. You inhibit another arm of a separate pathway, you get a cytostatic response. But only when you combine the two agents do you transform a cytostatic response to a cytotoxic one, synthetic lethality. So a few years ago, a graduate student in my lab, Alessandra Hillis, initiated in collaboration with Karen Czakowski and Steve Elledge here at Harvard, a synthetic lethal screen using anchor drugs, PI3 kinase and AKT, in a triple negative breast cancer cell line transduced with a whole genome CRISPR-Cas9 library, treating the cell with cytostatic doses of these agents, and then asking, these are barcoded, so asking the question, what genes, when depleted, transform a cytostatic response of the anchor drug to a cytotoxic one? And after gene set enrichment analysis and hallmark gene set analysis, we focused on one particular program of genes that were depleted in cells treated with the AKT inhibitor, and that is cholesterol homeostasis, which included the master transcription factors, regulatory elements of the cholesterol response, SREBP1 and 2. And another reason to choose this pathway is because there are drugs that are taken by about 120 million Americans every day for cardiovascular benefit, cholesterol lowering. And these are the statins, which inhibit the rate-limiting enzymes HMG-CoA reductase in the mevalonate pathway, which fuels the biosynthesis of cholesterol and the prenylation substrates FPP and GGPP. We focused on pitavastatin because it has the highest dose range and bioavailability. If you combine pitavastatin with both a PI3 and AKT inhibitor, two different structurally unrelated AKT inhibitors, you see synergistic inhibition of cellular proliferation on triple negative breast cancer cells growing in vitro. The same is true in primary patient-derived organoids. When you combine the two drugs, you get a cytotoxic Induction of cytotoxicity is measured by cleaved caspase-3 measurement. When you do this in an ER-positive primary patient-derived organoid, you do not see any cytotoxicity. Studies we conducted in collaboration with my colleague here at Beth Israel, Taru Maranan. And then finally, in vivo, again, the combination, the single agents give you a mild cytostatic effect, but it is only when you combine the statin with an AKT inhibitor in these in vivo experiments, you get cytotoxicity, a paper that we published about a year ago that if you're interested, you can see here. At the same time, these synthetic lethal CRISPR screens give you a wealth of information and potential other genes to follow up on. And Josefina Mendez, another graduate student in my laboratory, has been interested in mining the epigenome that has fallen out of these screens and focused on a BET domain protein known as BRD2. These are epigenetic regulators that recognize the acetylated histone tails, particularly those on H3 and H4. And by binding to acetylated histones, essentially initiate remodeling of chromatin to alter the transcriptional landscape of tumor cells. A large number of BET domain inhibitors have now been developed and are on phase 1, phase 2 clinical trials for a range of hematological and solid tumors. And when Josefina mixes potent BET domain inhibitors with either an AKT inhibitor or a PI3 kinase inhibitor, both of which are in the clinic, she sees potent synergistic activity, inhibition of cellular proliferation, and in the 3-day assays, induction of cytotoxicity. Again, only in the combination of the two agents. This is also true in patient-derived xenografts and in mice which are dosed with a xenograft where we have knocked out BRD2 using CRISPR-Cas9. And then finally, I want to turn to ovarian cancer. All of these are vignettes. I don't obviously have time to present everything. We turn then our attention to ovarian cancer. Why ovarian cancer? Well, it is one of those very lethal cancers that is a silent killer because patients typically present with either stage 3 or stage 4 aggressive, typically metastatic ovarian cancer, and they're either fit for cytoreductive surgery and then go on to adjuvant chemotherapy. And if they are not, then they undergo neoadjuvant chemotherapy followed by interval cytoreductive surgery. The other major reason is that this lineage is also littered with PI3 kinase AKT pathway alterations. This is an oncoprint that shows that there's PIK3CA, PIK3R1, PTEN, all 3 AKT genes are genetically altered. But unlike breast cancer, where you see somatic mutations or inactivating mutations or deep deletions in the tumor suppressors, in ovarian cancer, what you see are copy number gains. This is a copy number disease typified by microsatellite instability. So PIK3CA, there's a very few somatic mutations, the ones that you find in the same ones you find in hormone receptor positive breast cancer. What you see instead is copy number gains of the same gene. Same with PIK3CB, same with AKT1, 2, and 3, and deep deletions in PTEN and the PIK3R1 tumor suppressor. Ultimately, though, the consequence is exactly the same. You hyperactivate the AKT pathway. So ovarian cancer ought to be exquisitely sensitive to AKT inhibitors. Armed with this knowledge, Gil Saloom, a postdoc in the lab, conducted a second-generation CRISPR-Cas9 screen in a ovarian cancer cell line, dosed with cytostatic doses of a pan-PI3 kinase or pan-AKT inhibitor, asking the question, what genes are depleted against the anchor drugs? And instead of finding cholesterol homeostasis or many of the gene programs that we found in our triple-negative breast cancer screen, what Gil found is a gene known as ITGAV, which stands for integrin alpha V. It's an integrin. I'm sure you all know what integrins are. These are obligate heterodimers on the surface of all cells and tissues and organs, consisting of an alpha and a beta subunit. They are essentially responsible for binding extracellular ligand in the form of matrix molecules in the extracellular matrix. And this binding induces a conformational change, a clustering, which brings homo and heterodimers together, a conformational change which is transduced through the plasma membrane region, and the activation of downstream signaling pathways, which includes PI3 kinase AKT, but also assembly of the actin cytoskeleton. The importance of integrins was, as cell matrix and cell adhesion molecules, was recognized by the Lasker Foundation with the award of the 2022 Lasker Medal to Richard Hines Erkki Ruoslahti and Tim Springer. Now, it turns out that the Broad Institute had done all of this already. In their CRISPR data set of the Broad Institute, if you query the average gene dependency in epithelial ovarian cancer in their CRISPR data set for all of the integrin alpha and beta subunits, it turns out that the greatest dependency of all integrins in ovarian cancer is ITGAV, the very same integrin that fell out of Gill's screen. And if you then query the ITGAV gene effect across all cancers, epithelial ovarian cancer scores towards the top. And if Gill now takes small molecule inhibitors that have been developed and subsequently abandoned by a number of pharmaceutical companies because of poor efficacy and poor tolerability, if he takes these drugs and now combines them with PI3 kinase or AKT inhibitors, you see synergistic inhibition of cellular proliferation in vitro. And this is using both small molecule inhibitors and cyclic non-peptide RGD mimetics, shown here. And you can simply see by running a simple signaling experiment where you knock out the alpha V integrin with siRNA, or you inhibit it with a small molecule inhibitor, only when you combine it with a PI3 kinase or AKT inhibitor, do you see induction of cytotoxicity as measured by cleaved caspase 3 or induction of cleaved PARP, classical hallmarks of the apoptotic response. Whereas the single agents, either on the PI3 kinase AKT side or the integrin side, have little to no induction of apoptotic cell death. And when you do bulk RNA-seq of these conditions, not surprisingly, what you find is that the DNA damage response is the most significantly enriched program in cells treated with an integrin antagonist and a PI3 kinase AKT antagonist. So I've had time to share with you some of our efforts at Discovery Science using synthetic lethal CRISPR-Cas9 based screens across cancer lineages, trying to identify collateral vulnerabilities to PI3 kinase AKT inhibition that might give us combination treatment strategies that we can move into in vivo models and into phase 1 clinical trials, the work of Alessandra Hillis, Gil Saloom, and Josefina Mendez. I've said a little bit about our develops using and developing the next generation PI3 kinase and AKT inhibitors, work by Emmeline Leckie, Isabella Del Priori, and Chris Dennehy. What I haven't, unfortunately, had time to share with you is some of our other equally exciting work where we are targeting other cancer lineages, such as liposarcoma, and exploring alternative mechanisms of tumorigenesis and the metabolic vulnerabilities that exist within cancer lineages, such as liposarcoma, specifically protein lipoylation. That is work that is being done by a graduate student, Maggie Dreishpoon. And then finally, a very important and exciting piece of work, it deals with a separate targetable metabolic vulnerability in cancer lineages, the work of Tashbib Khan, who is interrogating the functional importance of glycogen metabolism, which has also given us a handle on biomarker discovery. And I'll tell you a little bit about that in my final slide. I constructed this slide about 5 or 6 years ago, sort of trying to think about where is the field going. None of these points, and this is of course my own personal view, none of these points had a check mark against them about 5 or 6 years ago. But now we have AKT inhibitors that have been approved for clinical use as combination therapies, albeit only in ER-positive breast cancer. But I would predict that additional approvals are going to be coming in 2026 and 2027 outside of hormone receptor-positive breast cancer. We now have allele-specific drugs. This is remarkable. We have PI3 kinase alpha allele-specific drugs, and you are going to be seeing AKT allele-specific drugs coming online in 2026. You will also be seeing coming online AKT isoform-specific inhibitors, which I think are also all of these with the goal of enhancing the efficacy and treatment options for patients whilst mitigating the toxicities that patients invariably display. Where we really have not gotten a handle of, as I think as a field, is we have been had a challenge in decoding location-specific function of AKT. AKT is localized at the membrane, in the cytoplasm, at intracellular organelles, in the nucleus, and we really understand very little about how all of this is regulated and its functional importance. The field is sorely lacking pharmacodynamic and predictive biomarkers to identify patients that qualify for PI3 kinase or AKT inhibitors for phase 1-2 clinical trials. This is a really big problem in the field and something that we need to turn our attention to in the future. And I think, well, once we identify especially predictive biomarkers, you're going to see a lot more of these drugs being approved by regulatory agencies. And then finally, something that I sort of touched upon at the very beginning, we know a hell of a lot about AKT, and there's good reasons for that. But we know very little about the other effectors of PI3 kinase that transduce a signal in normal physiology and in pathophysiology that are independent of AKT, and we need to turn our attention to those as well, I would argue. Final slide to thank the amazing team that I am privileged to have in my lab and whose work I've had a chance to highlight today. Our website, very importantly, the agencies that have funded our work over the last 25 years. And if anybody's curious, yes, this is my license plate on my car. So I am done. I will be happy to take a few questions. I do have a hard stop at 1, so we have 4 minutes.
Thank you so much, Alex. Great presentation, really insightful, and clinical research and phase 1 trials. So I'm going to go right into questions. Our first question is, what is the mechanism?
I can read them, Isabel. Okay. I can see them on my screen. I'll just read them in sequence. How much PTK1 regulates AKT? PTK1 is obligate, rate limiting for AKT activation. There is no mechanism by which AKT can be regulated and activated in the absence of PTK1. How are these molecules identified? If you mean the small molecule inhibitors that I have talked about, those have been through rational drug design and medicinal chemistry. That's as much as I can tell you. What is the mechanism behind the release of the payload? Good question. The polymer backbone does not interfere with drug release. The ADC is internalized through endocytosis. It is delivered to the lysosome, where lysosomal enzymes break down, release the polymer backbone, release the payload from the polymer backbone, and is delivered intracellularly to inhibit. We have determined this. What would you say is the role of RAS in the activation of PI3 kinase? That'll take about an hour to answer, but RAS is a potent and necessary effector. PI3 kinase is an important and necessary effector of RAS, but I think in the context of tumorigenesis, that has been oversold. I would argue that the primary function, the primary effector of RAS in the context of tumor progression is the MEK-ERK pathway, but there is a direct RAS PI3 kinase alpha interaction, as I said, as originally identified in the 90s by Julian Downward, and that is a very important mechanism. Scaffold functions of AKT isoforms. When you use a PROTAC, can you get a clue as to say that? Yes. And that is the concept of AKT as a scaffold, which has been very understudied, is exactly one of the projects that is ongoing in the lab to try and decode its function as a scaffold, as opposed to a protein that as a kinase that phosphorylates substrate proteins exclusively. There's a lot of questions here that I'm not going to be able to get to. Targeting AKT in endometrial cancer, it is absolutely being done right now. I can tell you that those drugs are being put into phase 1 clinical trials today. The role of PTEN in this pathway, it is a negative regulator. It dephosphorylates PIP3, terminating the PI3 kinase signal. Lose PTEN, which is what happens in cancer, you do not terminate the pathway. The pathway keeps firing. And that is the basis of its function as a tumor suppressor. Inhibitors targeting receptors, IGF-1, that's an understudied area, and I don't really have an answer for you. So my time is up. I'm going to have to leave the other questions. I apologize. If anybody wants to write to me with these questions, I'd be happy to reply as best I can.
Thank you so much, Alex. We really appreciate the webinar.
Thank you all. Have a great day
Bye.
and happy holidays to everyone.
Thank you.